Hi, good afternoon. My name is Nguyen, and today I'm very excited to share with you some of the research project that I've done in UNC Charlotte in collaboration with Carolina Healthcare System, University of Arizona. And the theme of all these project is about the highly adaptive biological image classification. So I'm fascinated by the biological diversity of life on Earth. A variety of life on Earth are fascinating. We have 8.7 million species, and we just one out of those many species. We actually have over 1 million type of insects, 600,000 type of fungi, and 300,000 type of plants. You can imagine the important biodiversity of our economic system and also our human well-being. So according to the 2010 Convention on Biological Diversity, participated in 190 countries in the world, they said that at least 40% of the world economy and 80% of the needs of the poor are directly derived from the biological resources. You can imagine anything happen to the resources is going to be affect very significantly to the world economy and people live around the world. So in order to study different type of light, we need to collect data. And we have a vast amount of image to analyze this diversity. And the U.S. National Center for Biotechnology and Images have a 20,000 terabyte worth of images need to study. So we do have a lot of this system, but having a huge amount of data, we also need automated method to study them. And so the image classification is a well-studied problem in computer vision, and we have other repository that consists of lots of different objects and different data set, and the ImageNet is one of the largest repository that consists of 14 million images on over 12,000 type of classes. But regular images are very different from biological image. So when biological image classification, we're going to looking at some image samples. These are image patches from big pictures, and you want to tell what kind or what class of these image is. And in order to do that, we feed the image sample into a thing called classifier. That can be done as a machine learning method to be able to tell the class sample of these images. So for example, here I show these are different pollen type, and each of them have a particular name. So in order to build the classifier, we go through a process called training. And so this is the process. We have some human provide some class label, so we know what the label look like. We feed it into a learning algorithm for the training process, and we finally have a classifier. And we can use this classifier to recognize different classes given the image patch. So labeling can be expensive. For everyday images, just like image of a dog or a TV or a car, you can use regular people to provide label, and these called crowdsourcing. And Amazon use it, Google use it to help them classify their image data. So that's great. However, when you come to biological images, for example, a different type of pollens, or you had to recognize between cancer cells, cancer image versus non-cancer image, we require highly trained experts. And most of them are biologists, medical doctor, or physiologists working in the field. So it's important to reduce the labeling effort for these experts. And that's what our research are focused on doing, is to reduce the labeling effort for these experts. So what happened when we need to reduce the training effort is that instead of having a lot of training sample, we now have fewer labels, right? And it's also important to learn how to provide the efficient amount of label to train a new class. And the new class is when you have a new imaging protocol, you set up different experience, you want to study a different type of species or different type of plants, you're going to have to collect training data for these. And sometimes it's harder to come by the new training data. So using limited sample, they have some drawback, and most importantly is the overfitting issues. So for example, if you have two classes you want to classify, and not assuming we have a natural boundary to separate them, if you have a lot of training sample, what happens is you can be able to come up with a learned decision boundary that are very close to the actual one. However, if you have limited samples, say if you have only a subset of the training sample, a few things can happen. Is that your decision now can move different places depending on the subset of the sample that you have. So this is not consistent, and what happened here is that it overfit to the small training samples. So these are not a very good option. So in our research, we focus on the highly adaptive classification. So we're able to use some of the things we learned before and adapt it to a new situation. And so we aim to reduce the effort from the expert to train this biological image classification. And we've done it in threefold. Number one is that we're able to integrate relative entropy to the prior psi distribution to regulate the training. And we've done that in the Sterile Detection project. Secondly, we utilize the spatial temporal distance among the unlabeled data to create pseudo-label. And we've done that in the insect identification project. And third, we develop an active search to acquire the most informative sample to give to the expert to label. And we've done that in the pollen classification methods. So today I'm going to share with you a little bit about these three projects that involve in all about building a highly adaptive classification system. So number one is Sterile Detection, and this is collaborative work between UNC Charlotte and the Carolina healthcare system. So why we have to bother detecting cells? Okay? So in the drop of blood, there is a range of white cells in your blood, in the drop of blood. Typically, any healthy individual would have from 5,000 to 25,000 white blood cells patrolling the bloodstream. If you have lower than 5,000, typically the case that you have a flu or poisoning. When you have increased number of white cells is also not so good. When you have from 25 to 50,000, it means you have a viral infection and your body create a lot more white cell to destroy those bacteria. Or you take in some drugs, some medicine, and it's also increase your white cells. And if your white cell exceed 50,000 in the drop of blood, there's a bad news. You have blood cancer. These are called leukemia, and a lot of people are dying from leukemia each year. So that are the big issues in measuring the number of blood cells in your blood. So now that we have our training process and we want to utilize the previously collected sample to feed into our training. So that we can just limit the number of label here. We're gonna leverage things that we learned before. So the question here is, how do you utilize the existing knowledge into a new class? So we look into this problem. So learning from existing class, we assume that we have label from before that we didn't have to collect, but it's existing previously. And for each of those class sample, you can train some hypothesis. So for each class, you train a number of hypotheses. It could be one or more. And then we put all these hypotheses sort of into a knowledge base, so we call candidate hypotheses. And then we're gonna transfer these knowledge into a new class. So we have a candidate hypothesis here, and we're going to apply it into a new sample that we have. And typically what we have is a series of hypotheses or rules that are fit well to these samples, but they are overfit. So they did not generalize well, or did not perform well on the data set. And so overfitting is one of the issues we're trying to avoid. So there must be some additional measure that needed for the training. So the measure that we use here is to apply a psi distribution into these image. So for each hypothesis, we're gonna apply it on a unlabeled image. So we have no idea how well it's performed, but we wanna try to get some measures back. So typically, after we apply a hypothesis, we had a binary image where indicate white pixel is cell and non-white pixel is the background. And we don't know how accurate these are, but we know they give us some response. And we can do that for multiple hypotheses. Each of them gonna give us a different response. From these, we can cluster the pixel into regions so that we can say how big the size of these objects are. And from that, we can build psi distribution. So these are some of the response psi distribution for my hypothesis. And so now that you have this psi distribution, we're gonna compare it against a standard one. So you have compared between the two distribution, and you can use relative entropy to measure the difference between two distributions. So the relative entropy is measuring the bit it takes to decode from one distribution to the other. So you can see this is a measure between the two distribution. So each of the distribution we're going to compare with a standard one, and I'm going to explain the standard one in a little bit. But for each of these, we're going to have a measure in response. And typically, the lower the number, the better. It means it is very close to the ideal or the standard distribution. So in this case, we pick the second hypothesis with the smallest rate. So how do we measure distribution P, right? And we can utilize some of the shape assumption. So in our object, we can assume the circular shape assumption. So we can measure this prior side distribution by manually measure the object size. So here, if you have a cell image patch, by 2 clicks from the user, you can measure the size using the radius. So you can do that for different type of cells, and then you can come up with a distribution, assuming it would perform a normal distribution or Gaussian. So that's how you can build the prior side distribution to compare with other distribution. So how do that perform? We collect a set of cells. So these are 5 different types of cells, and about 50 image that we collected, and we label over 2,600 individual cells. And we're gonna use a matrix to compare our performance. So if you look at the image and you have a cells, and the machine also tells there is a cell, that's when you have a correctly detected cell, or true positive, TP, in the green. In the red is when the machine say there is a cell, where indeed they're not a cell. So that incorrectly detected in red. And then the yellow is a cell that go undetected. So the machine did not know that there's a cell, why indeed there is a cell. So these are the measures we can use to compute the recall and precision. So recall is how the percentage of things that you detect correctly versus everything that you detect. And then the precision is how well your detection. You detect most of the cell while without produce lots of false positive or a lot of cells that are not there. And from recall and precision, you can build a single measure called the F score. It is the harmonic means between recall and precision. So our detection method can be measured by a single numbers here for F score. So we also compare that with other learning methods that are existing, and the one that we are built our framework upon. So the baseline is adaptive boosting, is a popular machine learning method. We also compare with a transfer learning method. These methods are taking previous knowledge from what we learned before and apply to new one, and this one is published as CVPR 2010. And then finally, we combine transfer learning and side distribution regularization. This is our method, and we published it in machine learning in medical images in 2011. So let's do some visual comparison. So here we have an image of natural killer T cells, and let's see how using only 10 training data labels, see how adaptive boosting perform. You can see red and yellow mean bad, green is good, green is true positive. So you can see a lot of false positive in this image. So the F score for that is about 40.43. Now with training, using task strata boost or transfer learning, you reduce a lot of false positive, but you can see the F score get a little bit better, but you still see a lot of yellow and red, true positive, false negative in there. And finally, using our proposed method, we have the higher F score, and we reduce a lot of false positive in the image. So the F score of 0.82. So to do it quantitatively, we select the number of training effort from 10 to 100. So these are the number of label that each learning algorithm receive to train. And we execute training and testing 30 times and take in the F score. So you have a mean and standard errors for each of these numbers. So you can see different training effort. So task strata boost using transfer learning, leverage previous knowledge, be able to improve the performance compared to adaptive boosting by some measure. As you see here, 21%, 5 and 9%. And by using our idea on the size regularization, we improve additional performance compared to task strata boost. So we increase by 17%, 14%, and 8%. And what was also really interesting is if you feed all of the training effort for ADA boost, you get about 82% in this case. And our method be able to obtain the similar performance using only 20 of the labeling effort. So we save a lot on the training data, so our method is more highly adapted to the new data compared to traditional machine learning or transfer learning method. So that is the first project. The second project, we look into insect identification. And these are collaboration between the computer science department and the biological sciences department at UNC Charlotte. So the biologists study ants, and they ask us to help them detect ant in their control environment. And so why do we study social insects? Some social insect can cause $billions of damages in terms of termites, and also destroying the ecosystem. You think about forests and the choice trees, so it could cause lots of damages. Some other insect like bee can pollinate crops and in turn secure our food source. So we want to preserve the bee population. And also different social insect like ant also represent a model for complex system. A lot of biology is interested in how ants interact with each other and how they form a complex system by very simple rules. So we help the biologists study those behaviors. So let's visit our training process. Last time we added the previous training sample, but now we know that we also have a lot of unlabeled samples. So these are just the video images that we have without having any expert to look at them. So these are called unlabeled sample. So the research question is, how do you utilize the unlabeled sample effectively? And the way we do it is that we propose a spatial temporal distance. And the spatial temporal distance can simply defined by a spatial location along with a temporal frame in the video. So if you're thinking, if you have videos, the distance not only between two locations in one frame, but also the location on the previous and next frame. So that's have a temporal component in 2 dimensions. So this location, we can calculate this location by using optical flow vectors, and you can be able to connect these locations based on their movement or they not move in the videos. And this distance typically is a low dimensional. Compared to the feature space that most of the feature were extracted from some of our subjects, so it's low dimensional. So in explain it in terms of pictures, if you have, this is one picture frame, and if you have 2 objects in them, these can see how we can compute them throughout the videos. So for example, this green object, if you detect it, you will give it a label. What we're trying to do is how do you connect it to the next frame. If the object move, you can connect using optical flow. And so you can see that all these objects can be connected together in the same way. So, and then there's object that doesn't move. So it's some image pass that doesn't move. We can use optical flow as well to create the connection between them like so.
Cool.
So using the connectivity among the unlabeled sample, we can assign label to unlabeled data. For example, using traditionally, if you use a feature space, and these are all you have an ant crop image right here, and these are all of the ant patch image that closest to this image in feature space. And so if you do like nearest neighbor, you're able to hopefully label it the same, so you can see all of these images have ants in them. The problem with that is sometimes you get a mixture of image that doesn't contain an ant, or images that are very noisy, so you couldn't see whether ants there or not. But if you use the spatial temporal space, what we propose, then when you look at the closest sample in spatial temporal space, you can see most of these ants are having the same label. So you can collect a lot of different unlabeled sample by having the expert only select one sample here. You notice they have Further away, we have a picture of non-ant, but that's the distance is a lot further, so you can restrict the distance and select only the sample that you have higher confidence. So how does that work well? We conduct the experiment in 3 insect type, and for each insect type we label about 6,000 sample for our experiment. And these are the insects taken from the 2 type of ant and 1 type of termites. So you can imagine in the biological experimental settings, ant will be in the containers, and you can see how they behave, and how you can detect how many ants are there, how do they behave, and so on. So our task is to make sure that we be able to detect all the ant in the image without detecting some noise. So these are the food and some other obstacle, or even the eggs. And so same way with termite, you want to be able to track and detect termite in the pictures without making mistake on other things that happen in the scene, like noises and the border. So what method do we compare it against? Again, the baseline method, adaptive boosting. We compare it again a transfer learning method. The features is basically our method, but we use the feature space instead of spatial temporal distance. So that's another version of the method. And finally, we use our learning spatial temporal space. And this is a published work at CVPR, WACV in 2017. So visually comparison. If we have an image of ant and we want to know where all the ant in this image. Adaptive boosting, the F score will be 28%. And you notice here the green box is 2 positive, but the red one, the blue one, the red one is 4 positive, the blue one is 4 negative. So you want to have a lot of green box and a lot less of red and blue one. So for transfer learning method, using 8 label only, we able to improve the F score to 45. The proposed method worked out to be better. We need to reduce lots of false positive. Why only have like 1 false negative right here? And the reason is hard to detect because the ants are so close to the edge, so it make it difficult. And for these, definitely a little bit noisy image and the machine making a mistake. But most of the ant would be able to detect in this image. So quantitatively, we conduct an experiment. And to train sample, we select the number of training sample from 4 to 256, double inside each of the experiment. And we execute training and testing 30 times, measure the mean and standard deviation of the area under the performance curve. So the ROC curve is you can think of is a performance curve. And we measure the area under that curve. So the higher mean better performance from 0 to 1. So again, very similar as you see from last experiment. Adaptive boosting have some measure here. Transfer learning be able to increase perform by some measure. Now for both of our different version of our features, using regularization on the feature space and regularization on spatial temporal distance, we'll be able to have significantly higher performance than other methods. And you can see here, training using 256 training sample, we're able to achieve that using only either 8 or 16 sample, and you achieve very similar performance very quickly. All right, so that's second project. The third project, we're working on the pollen classification, and we collaborated with the Arizona University, where they study bee and pollen, so they was able to have a lot of challenging data set for us to look into. And so this is the collaboration between us. So why do they classify pollens? And you can see down here, they have a shoe sneeze. So these are allergy patient, and be able to understand, look at the pollen samples and understand what kind of pollen they're dealing with, help them develop treatment for allergy. Also, pollen play a vital role for crops. There's a pollinated crop like wheat and rice. It need to pollen. So the importance of how the well-being of your pollen sample are going to be important for the crops. So let's revisit our training process again. So this is what we have so far. Now what were we asking here is what if we help the expert to find relevant sample from the unlabeled data. So these are the case where the expert didn't have to look at all the unlabeled data, but we, instead of looking at the unlabeled data and find interesting, most useful sample, to ask the expert, what is the label for these? So we're able to ask the expert, and at the result, we can collect more label sample in the most effective way. So the way we do it, we're gonna use some active learning search, active search method. So you imagine if we have lots of existing sample that we have been collected before from other experiments, and we can know some sort of existing rule among them. And now that we have a target sample, or saying this is a class we're interested in learning. So what happened is you can use transfer learning to transfer the rule over. Now, sometimes these rules are not good. Sometimes these rules are overfit and so on. So you had to figure out a way to improve it. And one way to improve it, to look at the sample right in the border. So you're gonna say lots of these unlabeled data are everywhere. But now you wanna focus your search only on the sample that close to your current decision boundary here. So these are hard classified sample because the classifier didn't know what class it belongs. It's right there in the So our method is focused on searching for these sample that are closest to our current decision boundary. You notice there are other sample that closer to some other decision boundary, but our method would ignore this sample. So we wouldn't consider it, and which is save time, because these chances are good, they're not part of the class that we're interested in anyway. So we ask the expert for this label, and for the expert put in the label, we're able to update our rule to make it a more accurate one. So that's how we go around and we have an active search method to find useful sample to train. So we test our method on the pollen classification problem. These are 9 types of pollen. I show 5 types here in this image. The image is actually quite small, only 1 millimeter across. So you can see these are under the microscope. You can see different types of pollens in here. For the experiment, we label 770 pollen grain. In procedure, we use threefold cross-validation to test and train. We use 6 type as an existing, 3 type at the target that we want to learn. And for each of the experiment, we run the test and training 50 times. Again, we compare with the usual suspect here, adaptive boosting at baseline, transfer learning at the strata boost methods, and our own is combined transfer learning and target directed sampling, which is the approach I just described. This work is published in WACV in 2013. So this is a performance chart, and you can see a different number of label sample from 0 to 250, and the accuracy is how well the classification method performs in terms of percentage of accurate. So you can see as you feed more label sample, all methods tend to do better, and that's expected for any learning method. The question is, when you look at how many sample does our method take to achieve similar performance at the transfer learning method. So you can see here, both transfer learning and adaptive boosting need all of the training sample at 250 here in order to maximize its performance at about 92%. Now our method be able to select most important sample first, so it reach high performance a lot faster. In fact, it only takes 50 sample to reach a similar performance at other methods. So we reduce up to 80% the number of training sample needed to accomplish similar performance. And you also notice why all of the method eventually reach a similar performance. Starting out, we already have high performance compared to the baseline. This is because we also learn to utilize things that we learned from the past to make our methods better. So. Now, in terms of more specific in the classification methods, there are a metric called confusion matrix to learn to evaluate the performance of the classification methods. And so with here, I show you the confusion matrix for this experiment. So you can see there's a number from 0.01 and higher is better. So this is 9.99 is really high accurate for the methods. And you can see there's some confusion between a couple of class here. Class number 4 is kind of confused between 4 and 5. So you see these two classes are hard to tell the difference. You can also see in class, these two class here is hard to tell because there's a lot of error come from the second one. So you can see there is some interaction or some overlap between different classes. But overall, the accuracy have been above 90% correct. And so we are very happy to see that we're able to help biologists to recognize some of these pollens. So for example, the output of our classification method is that we're able to identify the border of the pollen, also assign the class. So the class label here visualized by different colors, so different color mean different class. So you can see we're able to tell how many pollens out there and what kind of class does it come from. So the biologists study pollen can use it for the experiment without looking at thousands and thousands of images. One of the applications that we use is called pollen counting, and this is when you establish some experiment and you measure whether the machine was able to count all the pollen as accurate as an expert. An expert in this case is palynologist, is a biologist who study pollen. So overall, for each of the type, the error had been very low, automatically count versus manual count, and you can see very similar performance count can be between them. And so this saved a lot of effort for biologists to stand in front of the microscope to count pollens and conduct the experiment. So we're confident that the method will help to conduct more biological experiments. So in summary, our research aimed to reduce training effort of the expert when working on training the biological image classification system in 3 projects. The first project, we work on cell detection. We integrate the relative entropy to a prior psi distribution on the sample. The second project, we use spatial temporal distance at a measure to select pseudo label from unlabeled data to help with the insect identification. And number 3, we develop an active search method to select the most informative sample for expert to label in the pollen classification problem. Certainly this is not my work alone. I like to acknowledge my research advisor, Nguyen, and other collaborators that have helped us along the year, give us a lot of data and guidance. This is a collaboration again between UNC Charlotte, Arizona University, Carolina Medical Center, and funding from the National Science Foundation.